Hello everyone, welcome back to Dr. K. Prem-PRIMER video lecture series presenting by Dr. K. Prem, that's me. In today's lecture, we'll talk about restriction sites properties. What are restriction sites? Restriction endonucleases binds to specific sequence in a DNA molecule. That sequence is called as a restriction site. I'm saying again, restriction endonucleases binds to specific site in the DNA molecule. That site is called as a restriction site. See, restriction endonucleases bind to specific sequence of DNA molecule, right? That sequence of DNA is called as restriction site, right? Once the restriction endonuclease binds to restriction site, then it will break down the phosphodiester bond link between the two adjacent nucleotides of DNA molecule. What it does, restriction endonucleases binds to restriction sites, they are specific sites, and they'll break down the phosphodiester bond that links the two nucleotides of DNA molecule. So in today's lecture, we'll talk about properties. That's an important feature of a restriction site is palindromes. Restriction sites are palindromes. Palindromes means when you read from backward or forward, it sounds same. It sounds same, right? So this is what we will discuss in today's lecture. You see, restriction endonuclease, once they bind to restriction site, they break down the phosphodiester bond that link the adjacent nucleotides in a DNA molecule. That's within the restriction site. Within the restriction site, they'll break down the phosphodiester bond that links the adjacent nucleotides in DNA molecule. Restriction endonucleases cut DNA sequence that have twofold axis of rotational symmetry or palindrome. Restriction sites which are recognized by the restriction endonuclease has to be palindromes or twofold axis of rotational symmetry. As I told you that palindrome means when you read them from backward or forward, they sound same, right? Those sequences, restriction site sequences sound same when you read backward or forward, right? And what is twofold axis rotational symmetry? When an object rotates some degree, right, it reflects or it resembles the same. You see here, an object that repeat themselves when it rotates 180 degrees. Here I'm saying twofold axis rotational symmetry. The restriction sites are twofold axis rotational symmetry, means that sequence rotates 180 degrees, then it resembles its original sequence order, right? So I'm going to explain this palindrome or twofold axis rotational symmetry with the help of EcoR1 restriction site. As you all know that EcoR1 restriction site, EcoR1 restriction endonuclease restriction site is GAA-TTC. I'm reading from 5' to 3'. 5' means 5' phosphate, 3' means 3' OH. So GAA TTC. Again, 5 prime GAA TTC. So here the first strand where I have 5 prime is blue color and 3 prime is black color. In case of complementary strand, 3 prime is blue and 5 prime is black color. You can see the same sequence, this restriction site rotates only 90 degrees, then it looks like this. You see, if it rotates 90 degrees and 90 degrees, it looks like this. The same, and if it rotates one more 90 degrees, that's a 180 degrees, then you can see here the sequence. In original 5 prime, first strand, that's a blue color. Here you see the first strand, 5 prime is black color. But if you read the sequence from here, and you get the same, GAA-TTC. So the sequence, the restriction site, when it rotated 180 degrees, then it resembles its original structure or original sequence order. This is what is called as a two-fold axis rotational symmetry or palindrome. You can see here 5 prime is a blue, here 5 prime is black. But the order of sequence here is GAA TTC, here also GAA TTC. So the restriction endonuclease binds to sequence which are having the two-fold axis of rotational symmetry or palindrome. This is what is the important property of a restriction site, right? So this is all about small pep or a pep talk on restriction site. Hope you like this one. And if at all you try to subscribe my channel, that's a Dr. K. PREM-PRIMER. See you all again soon. Until then, bye-bye. Don't forget to subscribe my channel. That's a Dr. K. PREM-PRIMER. Thank you all.